How do you sample plants for the plant microbiome project? First, let's get to know our kit. At the bottom corner, you can find the kit number and a small scale bar next to it. In the kit, you will find a bilateral metal spoon, one side of which is a spoon and the other side looks like a small shovel. 2 pairs of scissors, one for cutting leaves and one for cutting roots, gloves, encased a bag to be also utilized for the disposal of used gloves, and 5 test tubes. The tubes contain a special preservative for the samples until they reach their destination in the laboratory. To get started, type the kit number in the appropriate line in the sampling form. Now look at the plants in the sampling form and go for a short walk in your surrounding area. Choose the plant you wish to sample and study and mark it. The choice is yours. Some of you will choose to study giant ancient trees and some of you will choose to study small shrubs or single plants. So the next step is very important. On your kit you will find a scale bar. Place the scale bar next to your selected plant and take a clear photo to demonstrate the plant size. Upload the photo into the corresponding place in the sampling form. Now perform a measurement of air temperature using a weather application and enter the result in the appropriate place in the form. And in order to let us know your exact location, pin your location using Google Maps. Check the location on the map and confirm the presence of your coordinates, a numerical representation of your geographical location. And then upload the photo to the respective place. We will start sampling. The first test tube is labeled background sample. Take the test tube labeled background sample out of the kit and open it. Pass the test tube between all members of the group, so as for all members to hold it in their hands for a moment. Seal the test tube and place it in the appropriate location in the kit. The second tube is labeled soil sample. First, put on gloves. Now position yourself about 1 meter away from the plant. To avoid contaminations such as footprints or animal droppings, we sample the lower layer of the soil rather than the top layer. Therefore, remove the top layer of the soil to a depth of about 1 centimeter. Use the shovel to take 3 soil samples. Place the samples into the designated test tube and seal it. Place the tube in its corresponding location in the kit and change gloves. The third test tube is labeled root sample. Expose some of the plant's roots using the shovel. Sometimes a lot of digging is necessary. Don't give up. Using the scissors, cut off a 2 centimeter slice from the roots. Place the root sample in the designated test tube and make sure that it is completely submerged in the liquid. The liquid in the test tube will keep the sample hydrated on its way to the laboratories at the Weizmann Institute. Return the test tube back to its location in the kit. For the next step, change your gloves. The fourth test tube is labeled leaf sample. The sampling should be performed on new, fresh leaves or leaflets, since these contain the microbiome we intend to sample. Use the scissors to cut a 2 centimeter slice from the tip of the leaf directly into the test tube. Place the sample in the test tube and make sure it is completely submerged in the liquid. Place the test tube in its location in the kit and change your gloves. The fifth test tube is labeled fruit or flower sample. Using a different pair of scissors or using your hands, cut a section of a fruit or flower and place them inside the test tube. Ensure that the sample is completely submerged in the liquid inside the test tube. Place the test tube in its location in the kit. Make sure that all the test tubes are properly sealed. Now, enter your contact details in the corresponding place in the sampling form. We may want to contact you to ask further questions about your samples or to keep you updated on exciting scientific discoveries that were made possible thanks to your samples. Do you have any thoughts you'd like to share with us? You are most welcome to post them here. From all of us at the Davidson Institute and the Weizmann Institute of Science, thank you for contributing to science. We hope you had fun.